This week we're going to be investigating populations, and there are a few key points that we're going to cover. First of all, abundance of a population, that is, how many there are, how a population can be distributed, the age structure of a population, and finally, the variables that affect population size. Now, population abundance is also called population density, and there are quite a few levels of this. Now, we can describe it in two ways, qualitative or quantitative. Qualitative, we talk about words like scarce, rare, up to infrequent, frequent, abundant, and finally very abundant populations. And the quantitative is where we actually measure with numbers. Now that's more like where we look at the number per square kilometre or metre or hectare, and we actually measure how or average out how many of a population you'd find in each one of those given areas. And don't forget, sometimes populations don't just go up and down, they also go in volumes as well. So when you have bacteria, we would actually measure them in how many would be in, say, a milliliter, which is 1 centimetre by 1 centimetre by 1 centimetre. So sometimes they can be measured in volumes as well. To measure populations, biologists have several options. Now we have something called a true count or census. That's where they literally count every single individual in the area and identify them. Now this is hard because you actually have to have a means of identifying each individual in that population. So you either have to tag them, so each of them has a number. You have to identify specific physical characteristics of that individual so that you can name each one of them. And realistically, for populations of, say, insects and that, it's not possible. So other ways to infer the size of a population is to sample them. And there are various methods that you're going to try out this week. Now you might ask, why measure at all? Well, number one, for a big picture, they actually use the stability of populations or the changes in them to determine the health of the surrounding environment. Australia's Great Barrier Reef is a good example of this. Right now they're indirectly measuring the populations of different fish species on the reef by monitoring the sounds that they release. You can actually tell between a healthy and an unhealthy reef by the types of sounds that the animals emit that are living there. So by measuring the sounds of these species and by monitoring how often they're making the sounds and how many sounds are being released, you can estimate the populations of the different fish species and see whether they equate to a healthy reef sound or an unhealthy reef sound. The other reasons we measure populations is to look for signs of destabilization. So in cases of koalas, we look for signs where they're becoming endangered, or in the case of invasive species, we look at signs where they are getting to the point where they're out of control and then affecting the surrounding populations of other animals around them. Okay, distribution of populations. When we look at a population, we don't just count how many, but we look at how they're distributed in an area. And there are three basic ways that this happens. We have a uniform distribution, where everything is uniformly spread across an area, not usually found unless they're affected by humans. We have a random distribution where populations will randomly spread over an area and try and spread out as evenly as possible. And then we have this clumped distribution here where species will clump together in certain groups in an area. Now, usually these distributions, number one, they'll change over time, and number two, they'll often be affected by the living requirements of those animals. So for instance here, for all you know, if you actually had an overlay map to see what's happening around this clump distribution here, for all you know, there could be, say, a river which lives in this area. Or it could be that you have lakes in this area or water holes, and the species are actually distributed around those water holes. It could be that within this area here, there is a large amount of predators, and so the species die out in those areas where there are predators, or the environmental factors might not suit them. So in the case of plants, if seeds might populate over an entire area where they've been blown, but they're only going to grow up in clumps where the environmental factors suit their living conditions. So there's enough sunlight, there's enough water, and there's not a lot of competition for the little seedlings to get going. So the distribution really does then indicate what environmental factors suit these species and what environmental factors, like here, do not suit the species. Now, if you have a look here, the distribution relates to a few things as well. There's also breeding seasons, so distribution will change over time depending on the season, whether or not they're having babies or not. Penguins will go into the sea and distribute and live quite lonely lives when they're not breeding, but they will then clump together to breed in breeding season. Then the females will then leave and leave the dads with the eggs. So when you actually measure a population, you've got to make sure you measure them at the right time, because if we only measure just the males when they're sitting on the eggs, we're really only looking at half the population. Like I said before, habitable and uninhabitable areas will affect distribution, and also social orders and behaviours. So some animals will only get together for breeding, some animals will only get together when they need to protect each other. Some animals will live in social groups, but only around, say, one key male or key female. So distribution relates to the living requirements, but it's not only the abiotic or non-living factors that will affect them, it's also the social factors that will affect distribution. Age structure is something that we measure within a population. So when we do measure a population, we also look at how many of the population are young or infant, how many of them are adults, and how many of them are elderly. And this relates to whether or not they can reproduce or not. So the young are ones that are below reproductive age. Mature adults are the ones that are at reproductive age. And the elderly, of course, are beyond reproductive age and cannot help to increase the population. All they can then do is help to protect the current population while the adults are busy trying to have babies. So when we look at these, we look at their age structure in any population to assess the health of it, because all three of these are indicators of whether a population is stable or increasing or declining. Now here, the first one here, when we look at this age structure, we've got the number of individuals that are below reproductive age, the number that are at a reproductive age, and the number that are beyond the reproductive age. And this is a population in decline because most of the individuals are actually beyond reproductive age. They cannot help produce more individuals for the species. There is less adults and even less infants, which means that there are less individuals coming through that can add to the population. So unless there's a big change in factors here, this population is actually on the way out. Whereas this one over here that looks like a pyramid is an example of a population that's increasing. So you've got a huge number of infants or young that are coming up the ranks, a large number of adults that are going to be adding to those ranks, and only a few elderly that are in decline. So this one is a stable population, or it could even be an increasing population dependent on infant mortality. So all of these individuals here that are below reproductive age, if there's a high death rate in infants or young individuals, it still may not mean that this is an increasing population. It could simply be a stable population. Okay, variables that affect population size. Essentially the big picture is this. You've got to look at your birth rate, your death rate, and your migration rate. And migration means individuals coming in or out of the area. So your overall growth rate of a population will be affected by the births plus the immigration rate minus the deaths plus the immigration rate. So if you end up with a positive, that means you actually have a population that's increasing in size, or a negative means you have a population which is decreasing in size. A stable population has zero growth rate. Now you might have come across in your readings something about closed populations. This is isolated populations where there can be no migration in or out of the area. So a closed population would be maybe like a fish tank where you don't add any extra species in. Nothing can get in, nothing can get out. Essentially you're born and you live and you die in that tank. Now, there are several factors that limit population size. Now, there are environmental factors and species factors, as you can see. Environmental factors you pretty much know about: your temp, your rainfall, your space, your food, other resources. All of these lead to interspecies competition, and if there's too much competition, the weaker individuals will die and only the strongest will survive and reproduce. Now, there's other things called species characteristics, so their reproductive strategies are going to affect how they survive, plus also the amount of parental care. And you will see in your reading something called R&K strategies. Now, this is relating to populations that put a lot of energy into reproducing but not caring for the young, or not. So the R strategy species are ones that put all their energy into making lots and lots and lots of individuals, like cane toads and rabbits. They'll put a huge amount of energy into reproducing, and therefore they're the ones that tend to populate very quickly when they come into an area. The K strategy species are ones that put a lot more effort into survival and caring and nurturing their young. So they basically have a very different distribution of what they put their energy into in their lifespans. The K individuals are more long-living individuals, whereas the R strategy individuals tend to focus on survival of the species rather than survival of the individual. Now, this week you're going to be trying and practicing measuring different populations, and we're going to do that through some indirect methods and direct methods using sampling techniques called quadrats, transects, and maybe mark and recapture. Now, you'll go into more detail in these in your Biozone books. They're just three different ways of collecting samples of population and then inferring how many individuals are in the area. Now, there are different ways to measure a population we call direct and indirect. Direct is where you can actually see the species and count them, and that's fine when they're nice and big and when they're out in the daylight, when you're out there observing them. But sometimes you have to have indirect measures of populations because there are some species that are very hard for us to actually see. So we actually look at things like the dung that's been left behind, the excrement, and that allows us to indirectly infer how many species or individuals are in an area.